We have reached the last segment of our discussion on homologous recombination, where we will discuss about some biological relevances of this homologous recombination apart from meiotic and mitotic recombination or repair. These are a kind of species-specific or organism-specific phenomena where the recombination, homologous recombination actually results in some exceptional features in those organisms. So we would discuss about mating type switching in yeast. Now, yeasts, they, as we all know, that uh, uh, they can multiply by simple binary fission, and then they can also reproduce by a kind of meiosis dependent sexual reproduction. So for that, not actually, rather there is a kind of sexual reproduction mimetic process there. So and that process is dependent on mating types. So there are two mating types in yeast. One is called alpha, another one is called A. So this alpha and A mating types, if you keep those alpha or A mating type in culture, often it has been observed that the mating types can be altered. So alpha can be transformed into A, and A can be transformed into alpha. Okay, so this can be observed in the yeasts, and therefore this is called mating type switching. This mating type switching event is associated with expression of a specific genetic cassette, which is localized in this loci called MAT loci. So here we have a certain genes which are, if they are expressed, then a certain type of mating type will be identified. So if in the MAD gene the A mating type set is expressed, the phenotype will be A mating type. If alpha is there, it will be alpha mating type. And then there are regions which are called HML, HM left, and HM right. So in this HM left and HM right, there are specific mating type cassettes that can reside over there. So it can be alpha or it can be A. Now, what would happen during this mating type switching? Because this both HML and HMR, HML and HMR, these are silent cassettes. So whatever genes that are there, those are not normally expressed. They are expressed only when these genes are uh, 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 actually positioned in this MAT loci through gene conversion. So there can be cycles where this gene in the HMR loci, it is positioned into this MAT loci, replacing the alpha genes through gene conversion, and replacing this alpha cassette that would result into mating type switching. So usually it has been observed that this kind of change, that means change of from HML to HML alpha to A, is quite frequent, and therefore this event involve some specific components, which are not the regular ones for your for uh, meiotic or mitotic recombination. And one such component is the HO endonuclease. So HO endonuclease, it can actually it can initiate this process of mating type switching by cleaving the DNA at a site which is nearby this MAT loci. The site is called Y boundary, and here the HO endonuclease can induce a nick, and subsequently the mat loci is degraded. So there is a gap over there, and this gap is filled up by aligning this segment with HML or HMR. So whatever gene copy is there, gene cassette is there, HML or HMR, that would be copied here, and therefore it would act, effectively this is basically a gene conversion event. So one gene cassette is converted to another gene cassette. Mechanistically, if we look into it with greater detail, you will see that if this is your mat loci, there you have the HO endonuclease targeting site. Now, what would happen when HO endonuclease is there? So it would induce nick in this particular position, and therefore it would result in the conversion of genes. So you can see MAT A would become MAT alpha. So here there is the nick in this particular site, which is the MAT site. However, in the HML sites, there is no nick because HO endonuclease do not induce any nick there. So here, after it induces the nick, there is a RAD51 mediated strand inversion, which is similar to the RECA mediated process in homologous recombination that we have already discussed. So this RAD51 mediated strand inversion would allow this part, the blue part of the DNA, to invade this red DNA by displacing one strand of the red DNA. And actually this displacement would allow the other DNA of the mat loci to act as a primer for DNA polymerase. So this will extend the DNA and this will copy the upper strand here, and the lower strand that has been kind of removed from this particular region, that will never be copied. So effectively what would happen, it would the top strand, due to this amplification, the DNA double stranded DNA will be synthesized, and in the bottom strand, after this replication of the top segment of the bottom strand, top strand of the bottom DNA, and the second strand will be synthesized after this. The red stretch, that was the mat loci single strand, after this is removed. So effectively what will happen, that the in both the DNA, only the alpha loci will remain. So this is a typical case of gene conversion that occurs in yeast, and that results in mating type switching. Among other biological relevances of homologous recombination, there is DNA damage associated replication fork restart. So when a DNA is severely damaged, there is stalling of the progression of DNA replication, particularly if the damage is there in the leading strand. So here the DNA polymerase, if this is the damage, the DNA polymerase is stalled over here and the replication would stop. Subsequently, the replication will stop. However, in some cases, the replication can initiate after this damaged segment, and we call it post-lesion DNA replication initiation. So through this kind of event, the DNA replication can actually restart. Another way to restart to start DNA replication here is associated with a recombinational event. So what happens here is that this partly replicated DNA, this invades the top newly replicated DNA, which is derived from the lagging strand, and thereby forming this D loop. Now, what this D loop will do, this D loop will come and this D loop will base pair with the damaged DNA strand. So when this D loop base pairs with the damaged DNA strand, then what would happen? The top strand will be copied by the invading strand. So some copy will be made over here by some DNA polymerase, and here you have another double strand. So these junctions will be resolved by normal recombination resolvases, and therefore this will yield two mature double-stranded DNA. One with the damage and the other is the normal one that should be derived from the lagging strand. So this way there can be replication fork restart following the DNA damaged lesions. So this way the recombinational repair can actually help the DNA polymerases or the replication process to overcome severe DNA damages. This is one of the important biological relevance of recombination. So now that we have discussed all the biological relevances, let us discuss some other importance or some other applications of DNA homologous recombination. So in laboratory, we use homologous recombination for several purposes, and one such purpose is gene replacement. Say we have a specific gene in one DNA segment and we want to replace it with some other DNA segment. So here we have, say, yellow fluorescence, so some gene YFG, sorry, not even fluorescence, yellow fluorescence, but some gene YFG, and we want to replace this gene with the antibiotic resistant marker. So what we would do, we would just incorporate the resistance marker flanked by the DNAs which are identical to 3' and 5' of this YFG plus gene. So it would recombine there and it would replace the YFG gene. So any target gene can be replaced by this, and this can be accomplished within a cell if the cell possesses a strong homologous recombination machinery. Similarly, for yeast, if you want to replace one particular gene, what you have to do, you have to have some marker. Like the URA3 is one of the markers for yeast. So if you want to replace one particular gene, this gene B, which is flanked by gene A or gene C. So just you should have URA3 gene within gene A and C, and therefore after this homologous recombination, this double crossover, the URA3 gene will be incorporated, will be replacing this B gene, thereby you'll be able to accomplish gene replacement. Then we can perform gene disruption here. So how gene disruption can be done? Say if there is some ORF, say here it is YFG ORF, you can have some marker like kanamycin marker. Now this kanamycin marker can be amplified with a primer where some extended DNA is identical to the DNA that flanks this YFG ORF. So now kanamycin is flanked by the 5' and the 3' end of this YFG ORF. So if you put this particular DNA fragment into a cell, it would recombine with the YFG loci, and thereby it would replace the YFG with this selectable marker. So by applying kanamycin or some other any other selectable antibiotic, we will be able to have the yeasts or other organism which are uh, where the gene is disrupted. Similarly, the gene disruption can also be accomplished by positive or negative selection. So just like we did before, we can use the neomycin marker, and the neomycin marker can be flanked by some homologous region so of in a DNA. So there can be a recombination to replace this region with a neomycin marker. And This particular organism can be selected by antibiotic like neomycin or G418 to have this knocked out organism. Now we can perform something more, that we can include this in this particular case set. We can include neomycin gene, as well as we can include thymidine kinase gene of herpes simplex virus. So if we can allow these two segments to recombine over here, we can have neo-R as well as we can have the thymidine kinase gene. Neo-R can be used for positive selection in presence of neomycin, and the thymidine kinase containing marker can be used for negative selection, where the presence of TK would activate some drug like ganciclovir or FIAU, and therefore it would actually mediate negative selection of the organism. So we can use homologous recombination for both positive and negative selection of specific DNA segments. So as I discussed before, that TSV, HSV, TK is basically used for negative selection, and NEOR is used for positive selection. So this is another application. Now we can use these gene knockout, gene replacement strategies in embryonic stem cells of mice, and we can culture embryonic stem cells in vitro, and we can actually introduce some of such constructs within the cell where homologous recombination will occur, and then we can select those genetically modified cells, gene disrupted or gene replaced cell, and we can actually implant those cells into some early embryo where these cells will divide, and therefore it would give birth to some mice with some genetic disruption. Subsequently, with some crosses, we'll be able to achieve uh, pure genetically disrupted mice with which we can perform several in vivo experiments. Okay, so this is how we can we can actually avail gene knockouts by homologous recombination. However, now we don't really depend on homologous recombination for generating knockout mice. We have some very well developed site specific recombination systems like CRELOX-P, and also now we have the CRISPR technology through which we can achieve genetic knockouts with a greater efficacy. But still, for some organisms, we still use this homologous recombination mechanism where this works fine in developing the knockouts. So we have discussed several aspects of homologous recombination in these 4 or 5 segments. So I guess we have conceptualized homologous recombination through this discussion, and maybe in our next segments we will discuss about site-specific recombination and transposition. Thank you for your involvement, your attention, and hope to see you soon.